You're listening to the Effective Statistician Podcast, a weekly podcast with Alexander Schacht and Benjamin Pieske, designed to help you reach your potential, lead great science, and serve patients without becoming overwhelmed by work. Today we are talking about patient-reported outcomes, or PROs, and the FDA patient-focused drug development guidance. A really nice interview with Rachel Lawrence. So stay tuned for that. Patient-reported outcomes are a really important thing. Everything is moving more towards patient-centric in terms of medicine. And I think that makes a lot of sense because patients are just much more experienced, educated, and want to have a say in treatment. And so patient-reported outcomes is a really important topic. And of course, FDA is a really important player. So listen to this really awesome interview with Rachel. I'm producing this podcast in association with PSI, a community dedicated to leading and promoting the use of statistics within the healthcare industry for the benefit of patients. Join PSI today to further develop your statistical capabilities with access to the video-on-demand content library, free registration to all PSI webinars, and much more. And of course, there is the PSI conference coming up in Gothenburg. So if you have not registered yet, that's a great opportunity to do so. Rachel and myself will be there, and lots of other people. So head over to psiweb.org and learn more about this, and see you in Gothenburg. And now let's go to the episode. Welcome to another episode of The Effective Statistician, and today I'm talking with a good friend, Rachel. How are you doing?
Thank you, Alexander. Very well, thank you.
Yeah, it's really great that we're talking together on the podcast. We have been talking to each other through PSI for a very long time, and have met quite often at all kind of different PSI related occasions. So it's great to have you on the show today. Maybe you can talk a little bit about your career up to now. What brought you here and what brought you into this topic of patient-reported outcomes?
Yeah, thanks very much, Alexander, for the intro and for the invitation to talk to you on the podcast. So I actually started my career not as a statistician at all. I did a biochemistry and genetics degree, and I started working at AstraZeneca as a molecular geneticist in the genetics lab. But actually, I found rather than doing PCRs, which everyone now knows exactly what they are, I liked analysing the data and understanding that. So I sort of moved into statistical genetics, and that's sort of what kickstarted more my statistical focus in the career, and then did a diploma in statistics while I was at work. And through working in the research area in genetics, I started working on the polymorphisms or the variations in the drugs that might lead to different responses to treatment. And we collaborated with the clinical teams on some of the projects with the clinical trial data to understand if that was indeed reasons for some of the differences we were seeing in treatment effects. So that was kind of how I got introduced into the clinical trial side of the statistics department at AstraZeneca, and I moved from the statistical genetics research area into the clinical trials section, and in terms of working as a more standard clinical trial statistician, where I met many colleagues and really grew and developed in terms of statistical application working clinical trials. So I worked in a respiratory disease areas on a few projects, and then I've mainly worked in oncology projects after that. So that was really good basis in terms of all the all the oncology endpoints and work we did at AstraZeneca.
Cool. When did the PRO interest kicked in?
So a bit of a mix. So actually, in my last project at AstraZeneca, I was the lead statistician, so I had to be really sure about all our endpoints. And I was worked together with colleagues in the PRO department on some of those endpoints, and realizing that they weren't very, we weren't very good at that time at really defining what we were going to do with the PRO endpoints, and that we just produced lots and lots of tables of descriptives and handed them to a different department, and nobody really worried about it. But I did work really closely that we really integrated that in those projects into the CSR. So I had that background in AstraZeneca. What really happened was AstraZeneca said they were going to move location down from where I live in the edge of the Peak District in the hills to Cambridge, and I decided that wasn't for me. So I left AstraZeneca, and then for a while I worked a little bit freelance with my own consultancy company while my children were still small, and that was the time I got more active with PSI, because I wanted to really keep connected to the community of statisticians in pharma, even though then I wasn't at AstraZeneca. And then after a while of working on my own as freelance, I really missed working in a team environment. And it was, I suppose, coincidental that I discovered there was a company right near my house that was looking for a statistician, and they worked on patient-reported outcomes. So it was kind of synergistic. They were looking for somebody to come in and work with a clinical trial clients on terms of that focus. So that's when I joined Delphi Values. That was about 4 years ago now. I can't believe that, time flies. And so since joining them, I've have a background in terms of clinical trials, really useful to apply that and learn the specialism in terms of patient-reported outcomes. And I think being able to have the two hats, so learning from experts at Adelphi in terms of the nuances and the things that go behind some patient-reported outcomes, make you appreciate the sort of richness of that data and how it can be used, but also can see it from the other side, from coming from the pharma side of what priorities can be in terms of perhaps regulatory or other, HTA or other audiences, if you like. So,
awesome.
Yeah, that's probably how I got to where I am today.
Awesome. Yeah, and PROs is really the topic, the hot topic for already quite some time. And there's also some episodes that I already recorded about it. And there's, of course, one stakeholder that is always important in the field, and that is the FDA. And so it's really great to talk today about the FDA and their kind of current view on PROs, as they have released draft guidance on it. But before we go into this draft guidance, maybe you can talk a little bit to kind of the history of how that evolved with PROs and regulatory.
Yeah, so I think as part of the FDA's efforts in terms of relationship with what they publish as their 21st Century Cures Act, the FDA sort of set out their mantra in terms of wanting to really develop some guidance in detail on patient-focused drug development, all the way from all aspects. So not just how we use it in regulatory decision making, but really what does that even mean in terms of collecting patients' input into the whole process, as well as development validation or fit for use questionnaires. So they put together a statement saying they were going to work on 4 methodological guidance documents to sort of address in a stepwise manner how to collect and submit that relevant data so that it could be used in the FDA decision making. And they want to touch on your recommendations for methods and as well as sort of analysis interpretation aspects. And I think that this started a while ago. So it was in 2017 that the FDA published their sort of intent to do these guidance documents. And there is an older FDA guidance document in terms of from 2009 in terms of PROs in clinical trials. But the idea is that the series of documents will, in the end, sort of supersede that and move things forward from the FDA's point of view.
Okay, yeah, very good. That's a nice overview. And by the way, if you listen to this and you're just thinking, oh, that's a lot of references, you will actually find all of these references on our homepage. So just head over to the Effective Statistician, where you can learn more about the references and also see what Rachel is up to otherwise, and kind of read her bio and so on. Okay, so in terms of these different guidances, where are we at the moment?
Yeah, so as I mentioned, the FDA started the process in 2017, and they did put a sort of timeline that these guidances should have been finished by the end of last year in total. I imagine that a global pandemic, has changed timelines. But as of today, guidance 1 had already been finalised, and actually, when we're recording this podcast, the guidance 2 was just been released last week, in the end of February. So they're kind of finalised and out there. Guidance 3 and 4 are still sort of on their website from the FDA as a draft format, although public discussion meetings for these happened back in 2019. So I don't know exactly when we're going to get these final guidance documents, but I imagine that they will be coming soon. But there's a lot of information even in the draft materials and the discussions that are out there that you can glean from the direction of travel and the sorts of topics that the FDA is going to have in these guidance documents.
Cool. So let's go into these guidance documents a little bit closer, one by one. And when we think about the first one, what's the key topics for the first one?
Yeah, so this is very early on in terms of collecting patient input in very general. So this is very much about thinking even at all about concepts and things that could be of interest to the patient. So it's a very, very sort of early on in terms of thinking about possible research questions and a methodology about how you could, who you're going to get input from that's relevant to that disease area. So whether it's patients themselves, whether it's caregivers, parents or carers in certain situations. So it's very early on in terms of thinking in terms of the questions that are included in that and the guidelines in terms of thinking about that patient input.
Okay, so this sounds like really kind of qualitative research that you would do at the beginning to kind of understand, okay, what are actually concepts that are important for a patient? Kind of, is it something around sleep, or is it something about kind of itch or pain, or what all the different things are that a patient might be concerned about for a specific indication? Yeah.
Yeah, and I think the guidance too goes into sort of, more of those methods for how you can get that elicitate that information from patients, how to do that qualitative research, how you do interviews and develop an interview guide, how you even think about selecting questions for surveys, how they consider that, to be applicable across different countries and demographics and sort of those sort of qualitative research topics. So yeah, guidance 2 has got a lot more information on those methods appropriate for that sort of qualitative research.
Okay, very good. So that is guidance 2. Interesting. That is. What do you think are the kind of main problems there with that need to be answered?
Yeah, I mean, I think actually, it's something I really learned a lot more when joining Adelphi. Had no sort of concept ahead of time of all the work that actually needs to go into sort of developing those questionnaires and finding out, what those areas are what even those concepts to start with are of interest, and how you go about doing that, and why it's important to do it very specifically for sort of your disease areas, or whether it's children or adults and things like that. So it's very important that we actually do really get those concepts really separated. It's a very big, it's very psychological. It's very much about all those sort of side aspects of, being understanding those very different nuances about what it is about. It wouldn't just be pain, for example. It could be something a lot more specific about, that interference, is how much it interferes with daily life, or how much can't you do, or where the pain is, the types of pain, or how prolonged your pain is, for example. And so where there are some, link concepts and things like that, in terms of really thinking about, really that concept elicitation phase is really getting to the bottom of those really important things through those interviews. And it's although it's qualitative and less familiar to as a statisticians, there's a lot of science behind how that qualitative research is done and applied, and it's a very rigorous scientific discipline in its own right, and sort of really underpins that what we might be used to seeing as a questionnaire turning up in a clinical trial. There's been a lot of thought behind that originally about how that's been developed. So and I think it's still an active field, and it still is worked on because of maybe the growing disease areas that we work in, or the different, making sure that concepts are appropriate across different demographics and that sort of aspect.
Okay. Guidance 3. What is guidance 3 about?
So guidance 3 is a lot more about, is how do you refine those concepts and impacts that the patients have sort of told you about, but how do you make that into a questionnaire, which we term an instrument. So in terms of how will you make sure that that those questionnaires, instruments that you give to patients are doing what you hope they do, that they're reliable, that they're accurate, that if you ask the same patient the same question lots of times and nothing has changed for them, that there's that you're getting consistent results, and equally that you are seeing change where you would be expecting to see change, so that they're sensitive enough to those relevant concepts. And I think this particularly sort of, the guidance doesn't sort of use a term like validation per se of all questionnaires and outcome assessments, but more that they're fit for purpose. But that doesn't just mean that they're sort of gone, oh yeah, I think that'll do. There's quite a lot of that's sort of the point of the guidance. There's quite a lot of rigorous about how you have to have demonstrated how it is fit for purpose, for that whatever purpose that is, whether that is for patient-physician interaction, or whether it's used in a clinical trial. There can be lots and lots of different areas where clinical outcome assessments are used. It's not just in clinical trials. So these whole sort of, the FDA is still interested, they could be used to help ascertain how you were going to use a medical device in terms of a walking aid or something like that. It's not all just about like drug treatments as well. So there's lots of different aspects that can come into those. So making them fit for purpose, I think is in guidance 3. It really, gives their kind of perspective in terms of those really important things that would enable you to say that and demonstrate that.
Okay. Well, you just mentioned a word or a phrase that is kind of related to PROs, and that is COAs.
Yeah.
Can you talk a little bit to see how these relates to each other?
Yeah. So I think that's when you're new to it, the guidance is suddenly saying COAs and you're like, hang on, I thought we were talking about PROs. So a COA stands for a clinical outcome assessment, and that's the general term for all these sorts of measures. So a PRO is a patient-reported outcome. So that does mean sort of what it sounds like, that the patient themselves report. But you could have caregivers helping, caregivers filling out a form, or parents, for example, filling out a form on behalf of patients. So that would still be a clinical outcome assessment as a terminology, but there are different acronyms depending on kind of exactly the type of questionnaire. So you'll kind of find in the guidance documents, they'll generally use COA as in their headings and those terms, but, PRO is a type of a COA.
Okay, very good. So in terms of these 4 guidances, what is guidance 4 then now about?
Yeah, so guidance 4 it is titled that it will, address the methodologies and standards and touch on maybe technologies to really think about how you're going to collect and analyze the COA data in clinical trials, or at least to incorporate those into endpoints that and then could be considered to be robust for regulatory decision making. And also to do that in guidance 4, there's also quite a big section on being clear about how you can identify what a meaningful change is in the clinical outcome assessment, because not necessarily a statistical significance is an important change for a patient. So there's that guidance that covers these sort of 2 sort of sections, I suppose.
Yeah, I think the important difference is a really, really interesting topic in itself, isn't it? Because when you think about it, an important difference can kind of depend on so many different factors. Yeah. So if you maybe start with a very, very severe disease, a small change might not, be relevant. But if you start with a very low symptom set, a small change basically means that you get rid of all the symptoms, which is probably relevant. Yeah. So this is one thing. The other thing is kind of change over time. Well, if it happens in a day, it may be really relevant. If it happens over a month, it may not be relevant. And this has nothing to do with p-values, because it shouldn't depend on sample sizes and things like this. So that's a really, really interesting aspect. What are your takes on this clinically relevant differences? Relevant for patient differences.
It's it is a big topic, and I think the FDA are very clear that they're very precise about being the importance of identifying what's an important within patient change. So almost as you're saying, for those patients, how much can they change? What's an important change for them? And that's some of the research that we do earlier on, potentially in the validation and looking at those sort of aspects. You can really, with the qualitative research, you can help understand some of those aspects as well, in terms of what would you say is, an important, change in these outcomes. So there's the focus on what's really meaningful and important for a patient, an individual response, and that's particularly what's focused on in the FDA guidance. And then I think there is also the aspect that, almost as you're touching on, there are different considerations for what's meaningful as a change over time. If you're looking at a group, so if a group of patients and their mean changes over time, what does that mean in terms of that interpretation of those mean scores over time? And also, how big should the difference be between the means between patient groups? So it kind of gets to start to sound all a little bit confusing, because you're like, well, there's 3 different types of changes that we might want to consider. But I think that's exactly why some of the guidance is there, and it's not just a simple one number at just this time point. You need to consider those aspects and that interrelatedness, and because of the depth of the patient level information, there's a lot to know. How many symptoms did they start with? How much have they gone down? Is there a floor and a ceiling effect? Because they can't improve anymore, but that doesn't mean there's not been some degree of treatment effect. So they all come together. But I think the FDA particularly, and in this document, are very clear about saying what's important for individual changes, and that can be useful also just to put into context. So that could be in the PROs, and you would like to say what proportion of patients, improved. That obviously depends on the threshold you've agreed for that change score to have moved by, but it also, becomes a quite interpretable number once everything underneath that is understood. So, how many improved, how many decreased, and we're basing that on saying they had to have changed by 10 points, 15 points, or whatever on the scale that's meaningful on that scale. And I think the thing that's important with meaningful change, and I think people do recognize, but it isn't just a blanket 10% change in a scale will suit everything. Questions are asked in different ways.
Not like with the IQWiG approach to it.
Well, if you have to start somewhere, IQWiG sort of like, right, 15%, let's use it for everything. And I can kind of see there's some, if you're not sure, you've got to start somewhere, haven't you? But I think that there's more recognition now that that's important. And I think this sort of guidance from the FDA of being a bit more precise and a bit clearer that those individual thresholds are really key will help.
Yeah, that's good. The other thing is it talks about technologies. So when I hear the word technologies, for me, that looks like we are talking about variables, or we talk about apps, or what's these kind of things. Is that what is in mind? Because technology doesn't look like you kind of fill out a questionnaire on paper. Yeah,
it's a little bit strange to be grouped, I found, in this guidance document, and it didn't have the draft didn't have a whole lot on this sort of aspect, but it was thinking about things like the impact, of, say, wearables to collect some of that information, or is it that it's, written down, questionnaire data and things like that. So there's a small section on reflecting maybe some aspects of different sort of technologies do you use, and even by technologies that sort of seems to have mean like paper copies for questionnaire versus electronic PROs, and those sort of differences, how you ask the questions can be important to understand the types of response you're getting. And it's not really technology, but sort of how that study design could impact it, when you're collecting those. So I think that's sort of what it was kind of getting at when it says it's going to address technologies. The draft didn't have an awful lot that was completely clear on that section. So I don't know how that's going to pan out in their final documents, and whether they're really going to detail a lot more about technologies, because technologies change quite quickly.
Yes.
So it feels a challenge in a guidance document that might sit around for 10 years. So I think it might just be a bit more of an outline of those sort of considerations of the sorts of technologies, and that what concerns they might have on how that impacts their decision making, because how robust or validated those are, not just the concepts that have been asked about.
Yeah. The next point I would like to talk about basically combines two hot topics in statistics. So that is really a super hot topic, so to say. It's a hot topic of PROs and the hot topic of estimates. So when we think about the kind of these two things that go together, what's the FDA's point of view on that, or what is kind of the draft guidance kind of going into?
Yeah, and I think that the draft guidance did sort of have quite a big section, and the way it was arranged was really taking the Estiman framework and splitting it and thinking about population, and really particularly a lot about endpoints, and then it links very obviously in my mind, sort of because it's about what is the research question, and fundamentally that's what the FDA guidance is about, is setting a lot clearer research questions, so that as a decision maker, a regulatory decision maker, you can then know what question has been asked and if it's been evaluated in a suitable way. So the draft guidance document was sort of pretty much based around sort of the Estiman topics in terms of the way it was organized. There was quite a lot of things in different sections, and of course I think in the Estiman, sort of using the Estiman framework a lot, it can sometimes feel like a little bit almost circular, because it is something a treatment, or is it an intercurrent or is it part of an intercurrent event strategy? If you're talking about, say, CONMEDs, is that the treatment allows you to have CONMEDs or not, or is it something that is then taking those CONMEDs that was sort of effectively disallowed? And I think things like that are really kind of key with a PRO type endpoint, where you could really see that taking anti-pain medication would potentially really influence if you were asking how somebody's pain was. So, it's maybe more so than some of the other endpoints. You really need to just think about, hang on, they're allowed to take loads of paracetamol in a study. That's no problem. But is that balance between the arms? Is it going to create a bias in your treatment effect when you're looking at a particular PRO? I think that's where it comes from. I think that the FDA definitely are using the Esteman sort of framework for framing this, and they had some examples in the draft guidance, but it was also before the Esteman ICH-E9 R1 was finalized as well. So I think I think the section might slightly change in language as everybody has been developing it in the last couple of years, and some of the examples maybe I think might get amended or altered or made more relevant in final guidance.
Okay, so it really talks about kind of treatment, population, endpoint, and these kind of things, really.
Yeah.
Okay.
Well, not so much the treatment in the current heading.
It's before that.
But certainly a lot about the endpoint. Yeah.
Yeah. Let's talk a little bit more heavily about the one aspect of it, that is how we actually then analyze the data. There are a couple of, let's say, analyses that maybe are especially suitable for PROs. And let's kind of go through these a little bit. So first that is mentioned is landmark analysis. So for those not familiar with this term, what does it actually mean?
Yeah, so it's interesting in the draft guidance that was almost sort of mentioned first, or interesting to me, because I don't know how applicable it is in every setting, but in general, it's in terms of, well, not in general, it's in terms of looking at the data up to a fixed time point.
Mm-hmm.
So I think I'm used to using landmark analysis more in a kind of survival oncology setting, whereas they're kind of using the same language to say, look, they want to look up to a certain time point. So the FDA quite often use an example of a sort of up to 12 weeks. I don't believe that 12 weeks is any sort of global standard. That's the only time we can look at PRO data, but it's always good to have an example. So, they're kind of saying that it's important to specify the time point up to which you're going to look at your analysis.
Yeah. Okay.
So whether that is then for a survival type analysis, where we usually typically think of a landmark, or but more generally they're using it to say looking up to a certain time point, which then could be applied with different, whatever sort of method you're looking up at that point. So I think that sort of guidance will stay, that just being a little bit clear for a regulatory endpoint, like just being clear about what time frame you're looking at.
Yeah. Okay.
Is useful for everybody and makes it a lot more comparable, perhaps to other treatments and things like that. And
okay,
useful from that point of view.
The second part is analyzing ordinal data, which I love personally, because I've worked on ordinal data already when I worked on my master thesis, or at that time point it was called diploma thesis. Ordinal data is, of course, something that you very easily get with kind of patient-reported outcomes, kind of.
Yes.
How do you feel on a scale from 1 to 6? Yeah, with 1 being, super happy, and 6 being kind of you want to kill yourself. So where are you on that? And then, of course, these kind of different labels, 1, 2, 3, 4, 5, 6, are ordinal in a way that, well, 1 is better than 2, and 2 is better than 3. But the difference between 1 and 2 is not comparable really to the difference between 2 and 3. And so what's kind of said about that in the guidance?
Yeah, and I think it's exactly that, the FDA is pointing out that many of these domains are ordinal and the models should be ordinal. We shouldn't necessarily just be assuming they're reasonably normally distributed and, are continuous. So I think, it's more thinking about, should we be presenting a little bit more descriptively in terms of percentiles or, and more sort of stacked bar charts rather than mean values. And I think it's about understanding the PRO instrument that you've used. So some can have quite maybe quite a range of scores. So although a single question might have just been scored on, 1 to 5, 1 to 7 scale, quite often some of the things we report, for example, pain, or even with the EURTC, even just the global health status, it's a couple of questions that have been used. And so the actual range of the values possible for a patient, it isn't just 3 or 5. Yeah. Actually it can range because it's usually a sort of multiple or addition, for example.
Yeah. Sum of items over, if you have 10 items, each range from 0 to 10, then you have
and I think
total score from 0 to 100.
And I think that it's maybe understanding from any given sort of instruments being used for that disease area, is it just kind of one question? And that's not usually that common in a fit for purpose questionnaire that's used in this way. They are usually a little bit more detailed. So you can use continuous sort of endpoint analysis. I think the point in the document is where it's not, though. And for one example might be in the EURTC, which is commonly used in cancer, is using things like the GP5, which is just like sort of how impacted are you on the side effect of treatments. And that is just one question, and we need to be looking at that. No, I'm not impacted. Yes, I'm impacted a lot. But lots of other things like pain, for example, can be a bit more continuous. So being clear which you've got, I think is what they're really saying. Do an appropriate analysis for the data you actually have, not just assume it's all the same.
Yeah, it's the same also with kind of, if you think about lots of the endpoints, they look into the COVID studies, like this OSCI, I think stands for, where you kind of go from being dead to being kind of completely free of symptoms and discharged from hospital. And so that's a typical ordinal scale. And whereas there's a big difference from going from dying or being discharged from a hospital. There's, by the way, if you're really interested in learning more about kind of analysis that takes these kind of ordinal structures into account, I highly recommend going back to one of the most downloaded episodes with Frank Konieczka about non-parametric analysis, because that's an approach that really just takes into account the ordering of the outcomes and not just kind of assumes some kind of other metrics on it. Okay, we talked already a little bit about time to event analysis with the landmark analysis. I think that's a pretty kind of clear thing to do. But what would be kind of typical events in PROs we would look into? Because it's not mortality or something like this.
Yeah, so I think we did sort of touch on it. It's related to what we talked about earlier, is that we would usually think about a decline in a score, a decline in quality of life, or an increase in symptoms that's meaningful to the patient. So it's a within patient change threshold. So you would be looking at saying, right, that a patient's had an increase in symptoms, that meets that threshold. So the time until, was that very quick, or was that delayed? And maybe more commonly, really the sort of more general sort of physical functioning or quality of life, that might be something that you're really interested in. Sort of that might be something that you sort of longer term disease that that you might be thinking, oh, my physical function is okay, it's maintained, but it's going to decline as the treatment effect declines. So you might be doing that. But equally, time to event also for improvement. Yes. Yep. So, when maybe there's tolerability early on with the study, but actually, then once you've been taking it for a while, your symptoms overall are improving and staying maintained, at that improved level. So time to improvement equally. So you need that threshold of change, individual change. That's key. In a deterioration setting, death sometimes also would be, regarded as an event, depending maybe on the disease area or stage of disease as well. So, and I think, the FDA in the document, it sort of says, depending where you are, death, consider death as an event as well. So, but I think the other thing that is sort of mentioned as well is that the censoring rules in a time to event analysis, are very important, as of course they are in any, in any analysis, and it should be made much clearer. There's always a concern about missing data, sometimes with the PRO data, and particularly perhaps sort of variations. Is a change, do you want a definitive change? Must you have got worse and stayed worse, or must you have kind of got stayed worse and stayed worse for at least 2 visits, for example, and depending how frequently you're measuring your PRO instrument and how long you're collecting post any other things that happen post treatment discontinuation or anything, that might influence understanding of how you apply censoring in your study as well. So there's the nuances to those definitions of the event. But I think the point really in the FDA guidance is being clear what the event is and what your censoring rules are. Sounds straightforward.
I think one of the key things for me in these considerations is really kind of how dense or how sparse you collect the data. Yeah. Mm-hmm. If you collect it only at baseline and endpoint, well, there's not a lot of time that you can do. Yeah. And if you kind of collect it maybe only every 4 weeks, but most of the actually differentiation happens within the first 4 weeks, then you will not be able to see anything. So these kind of things really, really play a role. So
and I think it's being clear depending on your disease area. Are you expecting to see a decline in quality of life in that in that disease area, in what you're measuring in that sort of time frame? If is it a one-off sort of injection treatment, is that sort of really something that then you're really interested in, a time to something happening? So I think it's it is used, but I think it's about, is the time to event the right thing with your data? What does it message? What question are you really asking?
Yeah. The next question is about responder analysis. So I think there has been written probably piles of paper on responder analysis and lots of kind of arguments about it. Oh, why should you dichotomize data when you have all the data? That is throwing away information.
Yeah.
Mm-hmm. Why is that in here?
I think it is because it has been very commonly used in PRO data, and because for an individual patient, it's useful to sort of think, did their change score, was that meaningful to them? So they a responder. But as I say, that sort of very relies on you've picked that one responder threshold, and then hopefully research that and you've got that. But you've already mentioned, as we've talked, that could be very different. 1 point change at either end of a scale might not really be the same thing. So I think that there's that aspect, and in the guidance, the FDA are basically saying in general they don't really recommend a responder analysis per se because of that kind of concern that you've collected this richness of data. But I think that what they do want to know is if you're then using this more, continuous data that we do analysis that shows that the changes that we are seeing are still relating to the ranges that are meaningful. So it's a funny statement in their draft document. They kind of talk about it and saying, it could be appropriate, but we don't really recommend it. Yet they also have sections saying it's important to think about individual change thresholds too. So I think it's that getting those together. They don't really recommend it as primary, but they are interested in understanding that changes are are meaningful. So
yeah, okay, and that goes to the last part that is mentioned, is the percent change from baseline, because that takes somehow into account that if you're starting with less symptoms and kind of you need to have a smaller absolute change to have something meaningful, versus if you have start with a lot of symptoms at the beginning and then have some change, so some improvement. I'm just thinking about the improvement scenario here. So what's your take on the percent change analysis?
Yeah, and I think that the guidance really sort of suggests that they're not keen on it either. Extreme caution should be exercised if you're going to use percent change from baseline analysis, which kind of possibly puts you off the idea of that being a really good idea. But I mean, they're highlighting this, almost concerns, you're saying, the floor and the ceiling, effects then becoming more of a problem because you can't improve over 100%. So yeah, they're kind of suggesting that's maybe not a great thing to do. That's my take.
Okay.
Yeah. But I think a change from baseline is fine and commonly used. That there isn't a section in the guidance document sort of being more positive on other approaches. So I think this section was more in the draft guidance. It is highlighting some of the pitfalls, particularly for PRO type data and why it's important to appreciate those in the PRO analysis.
Okay. Let's go to a last part, and that is about additional considerations. For me, that sounds like, and all the other things that we didn't know exactly where it fits, these are mentioned here. That's how I would set it up. So what are kind of the specific areas that are mentioned there?
Yeah, I mean, I'm sort of hoping that maybe when the draft document becomes more final, that they will find a home, because we've touched on some of those topics already. But they actually really do highlight a couple of things that we haven't talked about, that that they really are suggesting that for a COA to be really considered for a label claim, it needs to be clearly defined and also included in your sort of endpoint testing strategy hierarchy. So that concept was touched on elsewhere in here, but there's not any lots of detail per se in the draft document, other than making it clear that we should be able to be defining a PRO endpoint in such a way that it can be part of that statistical sort of hierarchy testing. Although, of course, as we've touched on, the fact that it's got to not just be a statistical treatment effect that we're looking for, it's also got to be a meaningful difference, is maybe more challenging to add to a sort of statistical hierarchy testing. But I think they're just pulling it out. Don't just sort of expect to put a PRO endpoint in and not do something that's robust to test that treatment effect.
Okay, it sounds pretty obvious for me.
Yeah, exactly. I'm sort of hoping that might come out more earlier in the document, because it's kind of saying you need to do that, and how do you get it in the endpoint strategy being clearer. And I think that's what they're also sort of suggesting in this sort of section of the document, that it should be very clear what the objective is.
Yeah. The other point also about kind of that it's clear in protocol and SAP. For me, that's also pretty kind of straightforward thing. Is there some kind of history that this hasn't always been the case, how PROs has been analyzed?
Yeah, exactly. I think it differs in sort of depending on therapy area. So where essentially a PRO has been kind of a key part of a primary endpoint has been done a lot better. I think, it's not more widely in lots of labels because it's been a secondary endpoint that's said it's not, we'll just look at quality of life or quality of life and symptoms. And I think the SMAN framework really helps. It is just being like, that's not good enough. I don't know what you mean by I'm going to look at quality of life and all the things we've just spoken about. I think that, there is those complexities and the subtleties, and it's not just, I'm just going to summarize a mean and then a mean across everything at every time point and then hope that somebody can conclude something from that. Actually, no, do you want to be specific about your time point, about which endpoint it is? Is it physical functioning, for example, that's really going to support your label claim, and other aspects that you've collected in the PRO more supplementary and support that. But for a label, they don't want to, they can't really be expected to think we're going to cover every single aspect.
Yeah. Okay. Very good. One last thing, there is currently a tick list in this draft guidance. Can you speak a little bit about this one?
Yeah, so they've kind of put a list of when you're planning a study, confirm lots of items. So I think it's quite reasonable. It's quite nice to have that in there, and it kind of touches on most of the things we've put before. Being a very clear endpoint. Suggest making it clear that it's what you're using is collecting meaningful outcomes, and that it's fit for purpose. That's sort of the validation. That your study design has been is adequate for collecting whatever it is you've set out to achieve. If you're looking at an immediate effect or impact of adverse events that are going to happen quite early on in your study, that you're collecting the data at the right sort of time, that the frequency and the timings of collecting those. They kind of seem obvious. Some of these seem obvious points, but that's they come together. That's what does make it a guidance document, so that everybody can see that you need all these factors. And I wouldn't say some of these are clearly not new. They are what happened, but I think that I guess in putting this together in their draft guidance document, they're pulling things out a little more clearly that maybe wasn't as clear for every sort of regulatory intent study. They're clear that you need to be the instruments need to be clear how they're going to be administered, and that people have been trained, or that the instructions for them are clear, and that everybody is going to be able to complete them. There should be, together with it being a well-known instrument, that, there are plans of how you're going to score the questionnaires, how they, as I mentioned before, sometimes the questions are combined. So, that's going to happen in line with how that's all been going to be done. I think it also mentions being very clear on, as we said, about putting that endpoint in the endpoint hierarchy testing, and any sort of plans for multiplicity adjustment if needed. And I think that might reflect on what type of PRO endpoints are relevant in your study and your labeling, in terms of what should be tested and how. Make clear plans on handling missing data seems fairly important, but
pretty straightforward. Yeah.
I think this is why it's in there, is that's the sort of thing that hasn't been as clear as it ought to be. The SMAN framework perhaps will help identify intercurrent events and separate out them from just missing data aspects. So once you know what your question is, that's a lot easier to write down what you're expecting to be there. And I think that's been one of the big blockers from regulatory perspectives when I've been involved in some of the debates, that it still seems to be the biggest concern, missing data handling. We're not sure about using missing data because using PROs because it has lots of missing data. And I don't know, from most of the studies that I've worked on, that in a clinical study setting, we try very, very hard to enable patients to be able to complete everything we've asked them to do, and it doesn't seem as much as maybe as a concern as it used to be in terms of missing that information. But it's still a massive sounds obvious, but it's a really big thing, I think, still, that we're really clear about what's missing. Because unlike any other endpoint, you can't just go and collect it afterwards. If it didn't get collected, if the patient was too ill to complete their patient-reported outcomes, you know, that's important to know. It's not just that it was missing. So there are nuances around that, I suppose. And yeah, the other things in the tick list are things like, you know, just being clear of how you're going to portray that treatment effect, the between group differences, and really, you know, how you're going to collect and store that data and from those. So there's a tick list. I hope it will become a little bit more of an organised tick list in a later document. But I think, you know, making sure you've covered all these sorts of things, all these concepts, even if they're in a different order, in my view, is really useful.
Yeah, that's good. And this was actually a really, really nice summary of what we really all discussed about. There's one last thing that I wanted to talk with you about. And I know this is already a pretty long podcast episode, but there's a new face-to-face event coming up, the PSI conference in Gothenburg. We have actually met at PSI conferences in the past, and I think we are both really big fans of PSI in general. So in terms of the PSI conference, for you personally, what are the best things about it?
Oh, yeah. Well, I'm really looking forward to going in person. I really hope in Sweden this year, because it's interactions with other people. So that, I think, is what I most look forward to. You know, maybe I came into PSI a little later in my own career than some who you know, been there a long time, but everybody's very welcoming and keen to meet new people. Obviously, I've been on scientific committee and part of the organizer for the conference for a number of years now, so it's really good to always meet the speakers and, you know, all these conference sessions that you've been planning or reviewing abstracts for, and you get to go and listen to the talks, which is much better than, you know, trying to just sort of glean things from abstracts and you know, understand only the details. So yeah, I really like that combination of meeting people, you know, listening to talks and then being able to discuss some of those topics as well. And the variety of topics is just really nice. It's an opportunity to go and to listen to something and listen to great presenters on a topic that maybe is outside your day-to-day work. And I feel you can always learn from how other people present stuff and how they explain things. Yeah, so it's a really good learning opportunity on lots of levels and great fun.
Yeah, you know what my biggest pain with the conference always is there's so much great content that I always have pain choosing which session I go to. Yeah, if you look these parallel sessions, I think like I want to go to all four of them at the same time, but well, unfortunately, I don't have kind of these magic things like the girl in Harry Potter. What's her name?
Hermione. Yeah.
Hermione. Yeah.
Going back in time.
Yeah, that would be really good. So, but I also completely agree. Kind of meeting also different people. If you've been there a couple of times, it feels a little bit like meeting friends from school again and talking to each other. So I highly recommend going there and already arriving on the Sunday, because the Sunday evening is already really nice to get together.
Yeah, and I do know for just to say, apparently it is a really, really nice city in Gothenburg this year. So everybody who's visited from scientific, you know, really said or live there say it's a really good city. So it's a really nice place to go. So.
Yeah. Awesome. So if that is your first business trip after the pandemic, that may be a really good choice. Yeah. Thanks so much, Rachel, for this awesome discussion about the FDA, PROs, and in the end about the conference. And I'm really, really looking forward to meeting you in Gothenburg. And all the best until then.
Thank you very much.
This show was created in association with PSI. Don't forget to register for the Gothenburg PSI conference. Thanks to Rain, who helps with the show in the background, and thank you for listening. Reach your potential, read great science, and serve patients. Just be an effective statistician.